Hey there, guys. I'm trying this again. I have no expertise on YouTube or video or on-screen acting or anything, but I kind of think like maybe I should give it a shot. So what I'm going to do is I'm going to try and give a series of little talks on things I have some expertise in. So I'm going to talk about the stuff I like to talk about at work all the time, and that is science, especially the science of developmental biology and evolution. So today what I'm going to talk about is genes, environment, and pattern generation. Pattern generation. What is that? That's this process where we build patterns, organized structure, where we have form and function all coming out of the development of an organism. So like you may notice my head is at this end, and I've got arms over here. That's part of a pattern. That's the pattern of the body plan. But I'm going to talk specifically about some patterns of regular order in an organism. And I'm cheating. So here in this video, I'm opening it up with a picture of Hox gene expression, because whenever anybody talks about evolution and development, they always think of Hox genes, for better or for worse. Hox genes are very cool. What you see in this picture on the screen is a series of different Hox genes that have been labeled with different fluorophores, different colors, so that you can see where they're expressed. So for instance, that red region, that's where ultrabithorax is expressed. And what you can see about this is that there's an array of these. There's a sequence of them from labial at the very front in segment number 1, all the way back and around and into the very tip of the tail where we find abdominal B expressed. Now I'm only showing this because I want to talk about a common misconception that all too often when lay people see this, the way they interpret this is that the Hox genes, they must be causal. The Hox genes must be creating these patterns. That the Hox gene is switching on to make that segment of the animal. And that is not correct. These genes will definitely specify subsequent development of these segments. That is, the identity of the structures that will form in those regions is determined by the expression of these Hox genes. But the location of Hox gene expression itself is actually dependent on an earlier set of genes that have been switched on to establish the pattern. So the pattern is set up first, then the Hox genes are switched on in that pattern, and then the cells in that particular region for a particular Hox gene are going to develop in a particular way. So it's not causal, it's a consequence. Now, this problem is compounded by another feature of the Hox genes. They are collinear with the body plan. Now, what does that mean? That means that if you look at the order of the genes on the chromosome, which is what that bottom line is all about, they're ordered from front to back there as well. So the front-to-back axis is specified from labial back to abdominal B. Let me get out of the way. There's abdominal B. And when you look at the fly itself, those genes are expressed in that particular order. It's as if the front-to-back axis of the animal is literally mapped in the genome, as if it's localized right there in the genome and shaped by the chromosome directly. But it isn't. That's not correct. We don't just translate gene order into body order. And those Hox genes are actually dependent on other genes that are turned on first. So there's a whole constellation of genes that come before Hox. Hox is not first. Hox comes on later. So for example, there are a set of genes that form stripes, segmental stripes. And this diagram is illustrating one of those genes. It's called Eve, which is short for even skipped. As you might guess, it's then turned on in odd numbered pair segments. There's also another one called odd skipped, which complements it. And there's also a whole bunch of others, things like that have names like Harry and Fushi Taratsu and These are all genes that are turned on in stripy patterns that are going to form the boundary for Hox gene expression. Okay, what turns on these striped genes, you might be asked? How do you make a gene come on in stripes? And the answer there is it's an earlier set of genes. So we go from Hox, we go back to these stripy genes, then we go back to another set of genes called the gap genes. The gap genes are genes that have broader patterns of expression than a single stripe, and they have their own little domains of expression. And one of the things the stripe genes can do is read the boundaries of these different overlapping genes and determine where they should form. In addition, as you can see in this diagram, you see all those little bars there down at the bottom that are pointing to things. Those inhibit the formation of stripes. Some of them induce the expression of a stripe. It's really, really complicated, all the interactions that are going on here. It's a spaghetti tangle of genes, all just kind of overlapping, and out of their complex interactions emerges a pattern, a very molecule-dependent pattern, but it's also dependent on who's talking to who and how they are inhibiting or exciting one another in order to set up these stripes. Okay, now I know what you're going to ask next. Okay, we go from Hox to stripy genes to gap genes. What sets up the pattern of the gap genes? And the answer there is there's another gene. It's called bicoid, and bicoid is what is called a morphogen. It is a molecule that it forms in a gradient from the front end high to lower at the back end. And what these genes do is they read that gradient of expression, and some of them get turned on where the level of bicoid is very high, and others get turned on where the level of bicoid is very low, and others get turned on when the level of bicoid is an intermediate value. So you can just sort of read this off and determine where you should be turned on, and that sets up the gap genes. Okay, are you detecting a problem here? That this sort of sounds like an infinite regress, right? It's just gene is turned on by a pre-pattern of genes, which is turned on by an earlier pattern of genes, which is turned on by an earlier pattern of genes. When does all this break down? It turns out it does sort of break down with bicoid. Bicoid is depending on more than just another set of genes to achieve its pattern of expression. It is depending on the physical structure of the ovary of the fruit fly, which is shown here. So we're looking inside the ovary of a fly. This is what is called a follicle. Inside the follicle is an oocyte. Okay, the oocyte is the egg that will eventually be mature and be pooped out of the end of the fly. Notice the structure of this follicle, that there's an oocyte, and then at the other end there's a set of cells called nurse cells. The nurse cells are helping that oocyte get very, very large. They're producing all kinds of gene products, they're producing proteins, they're producing RNA, and they're basically pumping it into the interior of the oocyte. So it's getting a big surplus of goodies for the baby fly to live on. It's pumping in things like, yolk proteins, so just, food for the embryo. But also it's pumping in informational macromolecules like RNA, like bicoid. So bicoid is being pumped from the nurse cells into the oocyte, and that's where this asymmetry becomes really important, because the bicoid RNA gets localized to just one end of the oocyte. So here's another case where we've got a physical structure that determines the localization, the pattern of gene expression, or in this case RNA localization, and that's going to lead to all this cascade of events that are going to occur in early fly development to produce a fly with a patterned body plan. Okay, so that's what's going on inside this ovary. One of the important points I want to make here is that normal development of the fly requires functioning genes and gene products like bicoid and also proteins that keep RNA locked into one end. But it also depends on a specific physical environment and an arrangement of support cells. All of these pieces, have to be in place to produce the cascade of interactions that lead to a fly. You need all of it. Okay, but that's a complicated example. I've given you a short rundown through the early development of the fly, which normally in a developmental biology class we'd spend a couple weeks probing, because it really is a complicated series of interactions. So let's talk about something simpler. Let's look at a cleaner, easier example that's maybe not as pretty. Let's take a look at your forearm. Go on, pull it up. Just take a look at it. Pull your cuff down. Just see what's going on there. I put a picture there of my forearm, but I'm sure yours is much prettier. I'm looking at that and I'm just thinking, boy, I got to start moisturizing. And look, there's gray hairs even there. It's getting old. Oh well. Anyway, so take a look at your forearm, and there's a pattern of hairs, and you may also see pigment cells. And if you look really closely, you may see things like pores. And you probably take this for granted, right? It's just coated with all this. There's also, of course, another pattern, which is that in your forearm, it's probably hairier than it is on the back of your hand, which is probably hairier than your palms, I hope. So there's this a super pattern there. But I'm just going to talk about the pattern within this localized area. Let's just look inside that forearm and ask what's going on. First of all, the pattern of hairs is not random. Yeah, you may look at this and you say, wow, that looks kind of sloppy. But no, it's not a random pattern. It's also not a crystalline pattern. There's different extremes here. You can imagine, well, we just sort of splat a bunch of hairs on there randomly, just throw hair starter positions everywhere. Or we could have some kind of very regular, very rigid model that specifies precisely the position of each hair before the hair forms. So we've got this array that could happen. But this is something in between. What do we mean by that? Well, what we mean is that there is no prominent patches, no really clumped up hairs anywhere. Also, there's no major bare spots anywhere. It's like we sort of roughly arranged all the hair cells to be distributed fairly evenly over the surface of the arm. We can even test this mathematically. If you measure all the distances from each hair to its nearest neighbor hair and then plot those together, you might expect to see a Poisson distribution if it's truly random. That is, you'd find some cases where hairs are really, really close together, and you'd find other cases where they're really, really far apart. And then there'd be an average where you find most of the hairs some average distance apart. When you plot the distribution of this kind of thing, what you find instead is not a bell curve, but a sharp but noisy peak somewhere at the median distance between the hairs. That's not trivial to set up. Another thing you can do is you can use, if you're really into mathematics, you can use an autocorrelation function to get a statistical measure of the period of the distribution. You can also do a power spectrum, all kinds of fun things like that, which I won't get into. But trust me, this is not a fully random distribution. It's also not a rigid distribution. It's something in between. Once you get familiar with this kind of pattern, you start seeing it all over in biology. For instance, in the arrangement of neurons versus glia in the developing insect nervous system, which I used to study, what you see is this salt and pepper pattern, but the neurons are roughly evenly distributed. They're not just sloppily thrown in there. And you probably, if you know anything about developmental biology, you may have heard about lateral inhibition and things like that. Models like that set up this fairly regular pattern. Another place I've seen it is I work on zebrafish, and if you look at the skin of a zebrafish, it's innervated by sensory fibers, and those sensory fibers branch differently in different individual zebrafish. They're not tightly specified genetically. There's a fair amount of variation there. But still, when you look at them, what you discover is that they get a roughly even distribution of sensory terminals, which again makes sense. If you're innervating the skin, the cutaneous sensory stuff, you don't want a big empty spot, a void where there's just by chance no terminals formed, and another one where you got a whole bunch of them, so it's super sensitive. You want it kind of evenly spread out over the surface of the skin. But again, you also don't want to dedicate so much genetic information to precisely specify where each one has to go. Okay, so we've got this evenly distributed arrangement of all these different things. Here's another example that I find really pretty. Look at these zebras. They're not all the same. There's no denying that there's a species-specific pattern. And you can look at these and you can say, oh yeah, I recognize a zebra stripe. And you can also look at details of the stripe and you can see that there's, like look around the eyes and you see this kind of pattern wrapped around the eyes. The neck is all these vertical lines. It's actually pretty cool that we've got this kind of pattern specified here. But at the same time, look really closely at them and you see they're all different. They have variations. Do you think those variations are because they're genetically different? I don't think so. I think this is just a chance distribution, that like those other systems I was just talking about, there's some variation that is only loosely specified. It says, make this pattern, follow these rules, and this is about how you will do it. But here we end up with zebras who have a pattern of stripes, a recognizable pattern of stripes, that is still as unique as what you'd see in a fingertip. That's another example. So look there. Look at your fingers. They end in fingerprints, right? And fingerprints are unique to every individual. They are not genetically specified. We know this because you can look at identical twins, and identical twins have different fingerprints. It's like the developmental system said, okay, we've got to have these stripes, we've got to have these whorls, we've got to have ridges and furrows. They got to be spaced roughly this far apart. They've got to have pores in certain places within that pattern. And then it tells the fingertip, go to it, just sort of follow these general rules and assemble it. It's not precisely specified, but there is a set of rules for the pattern. Okay, so we need a more tractable system, because I'm sorry, I'm not going to let you flay my forearms so that you can do experiments on the distribution of hairs in the skin. Zebras aren't particularly easy to work with. You can't tame them. They're big, they're bulky. They would probably kick you if you tried to flay them. And they're also expensive. So no, we can't do work on those. We got to find a different system to work on. So here's one. This might be familiar to you as well. So most of us have seen this. This is just a turkey, and some of you might be looking forward to seeing some of this next month for Thanksgiving. But here's what catches my eye when I see this. Look at those bumps. The skin is all bumpy. What's going on there? Those are feather follicles. So that's where, the poor bird was plucked first, but that's where a lot of feathers started out. And when you look at those bumps, you ought to notice what I've just been talking about, that it's kind of patterned, right? Those follicles, when you look at a particular region of the skin, you see they're a certain distance apart, and all of the follicles are roughly that far apart. And you don't see follicles piled up on one another, and you also don't see, at least within a particular region of the animal, you don't see great big bare spots. How interesting. How do they do that? How do they make this pattern is our question. So first, before I go on, if you've got an answer for that, and if your answer is there's a gene that does it, you haven't been listening. Okay, go back, start over again. There probably are genes. Let me make that even stronger. There are genes. There are definitely genes at work here. Those genes are probably plural, many genes working together, and they're working in complex ways to set up this pattern. And there are almost certainly other interactions with the environment that I'm about to describe to you. Also, I would say, since every single cell in that skin has the same set of genes, you ought to know that just saying a gene did it is inadequate. A gene interaction multiple genes interacting with their environment and their predecessors. That's the kind of thing we have to hear about. Okay, so there's got to be something going on. And the nice thing, unlike zebras and unlike me, we can do experiments on birds. In fact, birds are a major experimental model in developmental biology. The chick is just a classic system for studying developing organs in an animal. They're cheap, they're easy, they come in eggs, nicely packaged for you, and you can do all kinds of experiments on them. And since people eat them too, you can't complain too much if those experiments are done humanely. So I'm going to talk about one specific experiment that was done, and here's the paper in which that experiment is described. This is by Amy Shire and others out of UC Berkeley. I heard Shire speak this past summer at the Society for Developmental Biology meetings, and that's one of the reasons I'm talking about it now, is I thought it was a really good talk. I enjoyed it a great deal, and she talked about a lot of exciting and relevant things. So here's the paper. It was published in Science, and what they're looking at is what factors are involved in defining where feather bumps will form in an easily manipulated system, the embryonic chick. I'll give you the short answer first, and the short answer is it's a combination of genes. Yes, genes play a role, and environmental stressors. So yes, the environment is also important in specifying this pattern. Both components are necessary. Also, I have to say that the words emergent cellular self-organization, when I see those in the paper, title of a paper, I tend to read it. It's the kind of thing I really get into. Okay, so let's talk about the system. Here's the system. So chick embryos go from having a smooth skin at day 6, that's up there at the top left. It's just uniform skin across the surface of the animal, to 2 days later, at day 8, having a regular pattern of follicles. They don't have feathers yet, but they got follicles, and they form in a detectable, measurable pattern. So that's what you're seeing up there on the right at day 8. Down there at the bottom is just a photograph of what these actually look like, and you can get an idea. It's not quite as regular as the diagram on the top right, but it is, a regular pattern where you've got these roughly defined domains that are exclusive, and distances, there's a median distance between all of them. So it's a classic example of the kind of pattern I've been talking about so far. Okay, in these follicles, they have a molecular marker. That marker is something called beta-catenin. If you stain for beta-catenin, you also get this kind of distributed pattern that you see in that diagram. So one hypothesis early on was apparently, well, then maybe what's going on in these is that there's a pre-pattern of beta-catenin, that beta-catenin switches on in these little patches to form the feather follicles. And then the feather follicles form from that. But they looked carefully at it, and what they discovered is that no, that's not the way it works. There's no pre-pattern. That you go from the smooth skin to simultaneously having beta-catenin expressed and the follicles starting to form. So timing-wise, it doesn't work very well. So there's some mutual interaction going on there. So something else has to be causing these follicles to form. And you might say, well, maybe it's another gene. Maybe we just have to look deeper. Okay, if you're a molecular biologist, that's probably the way you're thinking is, okay, find more genes. Look earlier. We'll find the genes that are involved in this. But what they found is that it's something else. It's self-organizing in response to physical stress. So here's what they did. If you take that chick and you snip out a little patch of the skin, it will continue to develop in culture. You got the right nutrients floating around, and it's nice and safe, and it's not infested with bacteria. That little patch of skin will grow for days and days and days. So you put this in culture, and you just have it free floating there, so you've removed all the interactions with its environment except the fluid it's floating in. And what happens is, you might expect, is this little patch of skin just kind of contracts up, it wrinkles up and furrows up, and it fails to form the feather follicles. It just turns on beta-catenin everywhere, and it just makes this clumped mass. So it's got no pattern. That's the key thing. But here's the thing. You can then take that, little patch of skin, and you stick it down on an adhesive membrane. You just stick it down on a surface in your dish or your flask and just have it tacked down there. And now it's got something to adhere to. And so it can tug on that membrane and it can develop some physical stresses. And what happens then is it forms the feather follicles, the nicely distributed, evenly regulated feather follicles, if it's got some surface to tug onto. Okay, that's kind of cool right there. But then they go a step further. If you're tacking it down on this artificial membrane surface, that means you have the power to manipulate it. That means you could say, okay, well, I'm going to stick it down on a surface that's really stiff and rigid, or I'm going to stick it down on a surface that's kind of gooey and soft and flexible. And you can ask, what happens then? It's a natural sort of experiment, right? So they did it, and here are those results. So what you see in this figure is on the left are these little patches of skin that they planted down on a really rigid, stiff substrate. So this is a substrate that doesn't give much at all. The tissue is tugging on it, but it doesn't give, so it can't move much. And on the far right, it's on that gooey substrate where, again, it can stick, but it pulls and the whole substrate just kind of curls up with it. So it's almost as if it's floating in fluid. And then you got all the intermediates. And the cool thing is when you do this experiment, what you find is that, yeah, on the gooey substrate, same thing as floating, you don't get feather follicles. On the really stiff substrate, similarly, there's a problem with forming feather follicles. On the intermediate substrates, you get different densities of follicles. It depends on how rigid the substrate is, which is pretty neat. So it's not an all or nothing thing. It's like it's pulling on that substrate and generating degrees of tension depending on how rigid the substrate is, and that's influencing the density of the feather follicles that form, which is awesome. I like that. Now, furthermore, we're not done yet, because, as I said, what's generating the force is your cell is sitting there and it's tugging, it's kind of crawling and trying to stretch this membrane. So it's full of motor proteins that are pulling on this, and that's what's generating the force. The actual substrate doesn't generate the force. It's the interaction between the cells of the tissue and this substrate. So they took it another step further and they started manipulating the muscle proteins, the motor proteins that are involved in this, which is kind of neat. So what they've done there is, here's a similar experiment. In this case, what they've done is they've treated these pieces of tissue with either a molecule called blebbistatin, and blebbistatin is an inhibitor of myosin, so it just sort of shuts down the motor to varying degrees, versus treating it with another molecule called colliculin, which ramps up the motor, makes it contract even more, makes it grab even tighter to the substrate. When you do this kind of experiment, what you get is that range. These are all placed on a substrate that's identical, and you get this interesting range where, yeah, you get the distribution again, and it varies with how much traction the cell is generating. Okay, that's pretty impressive. That works really well. So it's saying that this is an environmental interaction. The other thing, I want to quote from the paper right there, because I thought this was a really important quote. So he says, the results are consistent with a model in which cellular contractility serves as a local activator and substrate stiffness serves as a long-range inhibitor of follicle aggregate formation. And little light bulb went on my head right there. I'll get to it in a moment. Thus, follicles emerge through a mechanical instability. So that's what we're looking at, is a mechanical instability caused by the interaction of beta-catenin with this substrate, that spontaneously generates an increase in morphological complexity. I wish there are a few intelligent design creationists who could get this, but yeah, that you can get increases in complexity that are not guided by an intelligence. It's a response to the environment. So my little light bulb moment that came on earlier, that's because of that next line. Of note, this mechanism bears resemblance to Alan Turing's models of chemical patterning in which a local activation competes against long-range inhibition, which is also really cool. So all you nerds out there know about Alan Turing, right? And in addition to doing computer science and cryptography and all this other stuff, Turing also dabbled in developmental biology, and he proposed these models for how you could generate patterns in simple systems. And one of these models is called a reaction diffusion model. It's a classic in developmental biology. And what this is saying is, yeah, this is kind of like a reaction diffusion molecule, although in Turing's case, he was postulating interacting molecules. In this case, we're saying it's interacting cells, and also that in this case, the long-range inhibitor is not a diffusible molecule, but it's the diffusion of the tension generated by these contracting cells on this substrate. So it's kind of neat to see. I'm always happy to see more confirmation of Alan Turing stuff. Okay, so let's look at a nice simple model diagram of how this all works. Yeah, that looks simple, right? Okay. The key here is beta-catenin. So that's the dark blue oval things that you see all over this diagram. Beta-catenin is acting as both a sensor, so it senses force, and it's also acting as a force transducer, that imposing stress on beta-catenin causes it to organize, and in particular to translocate into the nucleus, where it then acts as a regulator of gene activity. So you put stress on something, and beta-catenin responds to that physical force by entering into the nucleus of the cells involved and triggering new patterns of gene activation. So you, for instance, tug on this sheet of skin. In some places, beta-catenin goes in, and it triggers feather formation. So we're going to start making feather follicles there. Okay, so in this case, it's clearly a case for this mechanism to work. You need both things. You need an external forces resisting the cell, and you need a motor force or traction that can activate beta-catenin, and they've demonstrated they've got both of those. If the resistance is so high, as we see in the diagram on the far left, if you've got high resistance, the traction fails to compress the cells. They're just sort of futilely straining against the substrate. You get no localization of active beta-catenin. It's just kind of scattered all over the place. If traction is much higher than resistance, if it can tug a little bit on it, there's compression. If it's too much compression, the whole thing crumples in on itself, and you only get one localized region of beta-catenin, which is useless if you're trying to make a distributed pattern. But if you're in that sweet spot or that sweet range, that intermediate levels of traction and resistance, then you get localized distributed patterns of beta-catenin. Okay, so let me just summarize this because I've been talking too long. So a few words of wisdom I would pass along to you. First of all, if you see a newspaper article, or you never see science articles do this anymore, right? I hope not. If you see an article that talks about nature versus nurture, it's crap, right? It's a false dichotomy. In all cases, it's nature and nurture. Nobody is denying the importance of genes. Nobody is denying, well, some people are, but most people aren't denying the importance of the environment and how we set up interactions within those genes. In particular, if you talk to developmental biologists are all about how genes are talking to one another and interacting. I also want to throw out a buzzword I absolutely despise, and that's blank slate. I often hear people accusing others of being blank slaters and stupid things like this. Blank slate it's a straw man. No one believes that people are a tabula rasa and that there's no predispositions there. It's total nonsense to talk about a blank slater. It's also total nonsense to talk about uncompromising genetic determinism. We are not rigidly specified that we throughout our lives will have interactions that influence how our behavior works, how our physiology works, how we develop. And it's silly to ignore that as well. Okay, in addition to a genetic component, there are other things. Again, this is a layman's interpretation of how the world works, as you'll often hear them talking at length about, oh, it's all in the genes, that your genes specify everything, that it's just, you know, it's a cascade of dominoes. You knock over one gene and then it just follows through, and that's what you end up with. This is not true either. There are lots of components that influence how genes are expressed, and that includes chance. Pure chance does play a role in some things. There are historical components, and when I say historical, I mean both in an ontological sense, that during development, the descendants of a cell are inheriting properties that they acquired from their mother cells. There's also an evolutionary historical component, where how we develop, how we act, is affected by the changes that have occurred over evolutionary time. And there's also an important environmental component to this, as we just saw that in many systems, environmental influences are extremely important. And final point there, I would really like to get this across to everyone, that organisms evolved to respond to environmental cues. That we have a set of genes that have been tested over evolutionary time for their ability to be plastic and respond to the environment in all kinds of conditions, because it's very rare for an organism to live in a uniform environment over long periods of time. So all of us have evolved to be adaptable, and that's important, and it's really difficult to get that across to some people who seem to think that what they inherited in their genetic toolkit is everything there is to be about a person. Among the things that we forget here is that as human beings, education is a key factor in the development of the brain. And it's really silly to ascribe behavioral and intellectual differences to purely genetic causes. I'm not saying that there can't be genetic contributions, but to say it's purely genetic and that the environment isn't the predominant influencer of how your brain works is kind of silly. Okay, so I'm all done. Let me just roll the credits here. First of all, I gotta say who I am. I am P.Z. Myers. I am a biologist at the University of Minnesota Morris, which is a great little university located out here in rural Minnesota. I will say, yeah, send your kids here. If you're young enough, oh, hey, you don't have to be. You can be any age and get an education. If you feel like it, come out here and get an education. Send your grandkids here if you're in that generation like I'm getting to be. It's really is a good place to live and to work, and I've been very happy here, and I think our students have been very happy here. But I do have to say one thing, despite that advertisement I just gave, the opinions expressed here represent my own and not those of my employer. Standard disclaimer. Except I'm pretty sure they would agree with me that you should come here, that this would be a good place to go. That might be the one point we agree 100% on. Okay, also, I have a blog. I blog at freethoughtblogs.com, which is a network of other writers. I am not alone. I am also influenced by my environment, and this is my environment. You should go here and take a look at these people too. We have a lot of writers here. We've got a few scientists, so I'm not the only one. There's a few scientists working here. There's a heck of a lot of godless atheists here. That's one of the things we look for is people who aren't beholden to a dogma. And most importantly, the one criterion that is most important in selecting people who write here is that all of these people have a commitment to social justice and equality. So it's a good group of people. I like hanging out with them. You should hang out with them. Come on by, browse through our list of terrific blogs, and pick someone to be your favorite author here, because you got a lot to choose from. Okay, lastly, I'll just mention where you can find me. My blog is, as I mentioned earlier, is called Pharyngula. which nobody can pronounce and nobody can spell, but that's okay. I like it. It's the stage of development I find most interesting, so that's why I called it this. You can feel free to stop by here and leave comments. You can also leave comments here on YouTube down below, but I will say I have a comment policy here because you gotta admit this. The YouTube commentariat is a swirling cesspool of trolling turds, and most of them have nothing of any worth to say. So I've set my comments to require approval before they appear. So yeah, leave a comment. If it doesn't appear right away, it means one of two things. It means either you're an obnoxious troll and you should just go away and I don't want to talk to you. Or it could mean that simply I haven't gotten around to checking the comments queue that day. And if you wait a day or so maybe it'll appear. Okay, what else do I need to say? I think I'm done. Yeah, I'm pretty sure I'm done. So thanks for listening. I'm going to be talking some more about science in the near future. There is a subscribe button down below, so I'm not big on that, but if you want to, go ahead and subscribe. I have plans to have a weekly science talk here on YouTube, and right now what I've got in mind is next week I will probably talk about, ooh, this is going to be fun, development of the reproductive system, which will be very annoying to some people who have some very narrow perspectives on how that works. But we'll get some good controversy going and people will communicate with me and tell me interesting things, and all the trolls will get thrown into the trash bin. Okay, thanks very much. We're good. Talk to you later.